This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, a special episode recorded on December 11th, 2019. I'm Vincent and you're listening to the podcast all about viruses. Continuing on my Singapore road trip, today we are at Duke National University of Singapore Medical School, and I have a few faculty members from here and also a nearby university. We're going to talk about their science and their careers. We're going to make Singapore science famous. Oh, it's already famous, of course. We're going to help. So let me introduce my guests. These lovely red chairs, aren't they great? Really nice, thank you for the red chairs. Ashley St. John, welcome to TWiV.
Thank you.
You're the easiest name I have to pronounce.
That may be.
Thank you for that. Next to her, Sheemei Locke.
Thank you for inviting me.
Welcome to TWiV. And did I pronounce it well?
Yes.
Sheemei Locke, okay, thank you. And here's the most challenging. Eng Eong.
Well done. Thank you for having me.
Thanks for coming. And now at the end, from Nanyang Technological University, Dahai Luo.
That's right.
Luo? Or
I have a simple version with hi.
Thank you for coming. All right, so 3 faculty here at the DukeNUS and 1 from NTU. And I want to talk first a little bit about your careers. I want to know where you're from and your education. Let's start with you, Ashley. You're somewhere in the U.S., right?
Yes, I'm from the southeastern part of the U.S.
Southeast, that's like Florida-ish?
Actually, I was born in Atlanta, and I grew up in South Carolina. And then I moved back to Atlanta for my undergraduate degree at Georgia Tech in Atlanta. So
that's just there.
You were just there, yeah, I know. I
did a podcast at Georgia Tech.
I saw that. That's great.
It's a great campus.
It's very nice. It's improved a lot since I've been there.
Yeah.
Yes.
So you were an undergrad there?
I was an undergraduate there.
And were you a science major?
I was a biology major, yeah.
And then what did you do after that?
So then I moved to Duke for my PhD. I was interested in virology, and I worked for a while as I was an undergraduate at Emory University as a technician in a lab, and I got really excited. I was working in an HIV lab, and I thought, there's this other side to virology where you not only see what the virus is doing, but how the host is responding, and I got very interested in the complexity of that. So I applied to the Duke Immunology program and did my PhD there.
Who was your PI?
So I actually, my PI was Soman Abraham, who is actually a bacteriologist. So I, was given the advice to really choose the best lab for you, the best fit where you think you'll get good mentorship. So I did that, and I ended up studying salmonella. I studied mast cell responses to salmonella. So he has a lot of expertise in mast cells in his group, studying both allergic responses and responses of mast cells to bacteria. And then so I finished my PhD in, I guess, 2010 there.
And what did you do next?
So at that time, DukeNUS was just starting, and it was very intriguing to me that this department was starting in virology. I always had this passion about virology from before, and I wanted to do a postdoc where I could transition to more of viral immunology as a focus. And at the time when this program was starting, Dwayne Gubler was our program director. He's an expert in dengue. And I thought, since I haven't lined up a postdoc, maybe I will just go there, learn a little bit about dengue, and move on to another place pretty quickly. So I'd never really expected to be here so long. But one thing led to another, and I did learn a good bit about dengue from Dwayne and from Eng Eong and other people here in the program in Singapore. And then one thing led to another, and I applied for the PI position that opened a few years ago.
So I guess being at Duke, that helped because you knew about the university that was
Right. So it was really just starting. I think the building was just opening, that most of the desks were empty, and it was a super exciting time where everyone was very passionate about building the program. And I got I thought that was a good environment to learn about virology with all of these great scientists here in Singapore that study dengue. It's not as common in the US, so it's a big problem here.
I notice a lot of people work on dengue.
Yes.
Which it makes sense, local issues, you want to work on them. Not many people in New York City, for example, although there are some. And do you remember how far back you wanted to be a scientist?
Oh, that's a good question. I think I almost always wanted to be a scientist. I was always interested in science as the primary intriguing thing. I was interested in marine biology. I was very excited about this book that came out, The Hot Zone, about Ebola. I thought it was very fascinating how viruses are such a formidable enemy for us. And I think it was always something in the back of my mind that I wanted to study how biological systems work at some level.
It's interesting. Many people were inspired by the hot zone, even though it's half fiction.
Right. We know that now, but I think it's very valuable for the place it serves.
The way I look at it, if it attracts people to virology, it's okay. Sheemei, where are you from?
I'm born in Singapore, and I've been forever Singaporean. So when I was little, I'm very playful. I only like to play. And I do well in only the subjects that I like, math and science. And so I did okay. And then when I did my GCE O-level in Singapore, which is like an exam that you take when you're 16 years old, and my scores are not that great that I can go into the top schools, but I went to something called Ngee Polytechnic. It's like a university, but it's specialized in some courses. And I liked biotechnology, so I give it a try. And wow, it was so interesting because you can see why whatever that you learn, how you would translate into something practical, like plant tissue culture, how to grow a plant from a single cell, and how to induce the plant to flower in the right time for Valentine's Time Day. And I really liked immunology, is to study the immune, how your body fight against pathogen. And then after that, I did very well because everything is science. And then I went to Australia, Queensland, University Queensland of Technology, and I did an honours degree with someone called Dr. Jonas Skov, and he's the director of the Arboviral Reference Lab. So I studied how antibody can kill dengue viruses, and it's very interesting. So then during that time, I was trying so hard to map how, so the way we map how where the antibody bind to the virus is by just keep growing the virus in the presence of antibody and until you get an escape mutant, and then we sequence that, and hopefully the mutation is right at the site where the antibody binds. So I've been doing that for like years, and it's so tedious. And then Felix Ray, I think you interviewed him. Yeah, and he and Steve Harrison's lab actually solved the crystal structure of the tick-borne encephalitis E protein, which is very similar to dengue. And they can see everything. I was like, oh, what the hell am I doing here? So I should learn crystallography. So that's what I did. So I came back and I did crystallography, although working on an entirely different system in some snake venom thingy. And, but I want to return to cryo-EM. And during that time when I was doing a PhD, Michael Rossman and Richard Kuhn in Purdue solved the entire virus structure using cryo-electron microscopy. So it's even more powerful than the crystallography. So then I applied to Michael Rossman. And I was in Purdue for 4.5 years. It was postdoc, yeah. It was very exciting. Every day is very stimulating, and I learned so much from him. He's a really good scientist. And but it was quite, personally, is quite difficult because I was paid so low. My husband refused to come with me, and so my family stayed back in Singapore. So it's science is exciting, but the personal life is not that great. So then I decided, and my husband refused to move anywhere. So I have to come back here. So at that time, the department just started, and they see that I am a valuable person that can contribute to the department. So I think I'm hired. And I'm now a full professor after 10 years.
Great. Rossman was amazing. He just died last year, right?
Yeah.
Unfortunately.
It's very sad, yes.
He was awesome, yeah. So Felix Ray, I just released that episode last week.
Yeah.
So I know a little bit about dengue structure.
I know. He is my idol. We're collaborating now. So.
With Felix, yeah?
Yeah.
So he was in New York and he came to my office and we had a little conversation. He was great. Okay, Eng Eong, where are you from?
So I was born in Malaysia, north of Singapore.
Okay.
Came to Singapore when I was 12 years old and did the high school education here. And then went to medical school in the UK, at the University of Nottingham. So I wanted to become a doctor since I was about 14 years old. And I loved medical school, loved the subject. Came back and practiced for a year and I found it actually quite dissatisfying, especially medicine. I mean, the paradox is that I really love the subject, but in actually doing it, I found that it was not satisfying. It took me a while to figure out that I didn't like it because I couldn't solve anything. You're just patching a problem, but you're never getting to the root of the problem and getting rid of the problem. And I felt that the only way to really make a contribution to medicine was to really understand the science. And so after that year of practicing, I got into a PhD program at the National University of Singapore. And like Ashley, I chose the mentor. So my PhD was actually on nasopharyngeal cancer. It was nothing to do with virology. And I was quite open to the idea of doing cancer research, except that when I finished my PhD, Singapore had at that time the largest outbreak of dengue in history in 1998. And there was no one working on dengue. And no one understood why Singapore experienced dengue outbreak, even though it spends about $60 million to $100 million a year controlling the mosquitoes that spread dengue. So that was my introduction to dengue. Why did Singapore experience this outbreak? So I spent the first few years trying to understand the epidemiology of dengue in Singapore. That then got me into the public health system, and so I left the university, went into the public health system for about 7 years or so. And then when Duane Gubler came to Singapore to set up this emerging infectious disease program, and since I knew Duane and what he did, naturally this was the place to be. And so I was the fourth hire in DukeNUS. Mariano Garcia Blanco from Duke was the one that started it all. He hired Duane to become the director. Subhash was just there, was the third, and I was the fourth. So we were and I still have this picture of a retreat of the four of us in my house trying to plan the program in emerging infectious disease. So it was fun to be able to set all of this up. But since coming here, since we understood what are the issues with epidemiology and why Singapore had this re-emergence of dengue despite mosquito control, I became more and more convinced that we needed a vaccine. And so then the stumbling block for vaccine has always been pathogenesis. We don't understand pathogenesis, and that's why it makes vaccine development much trickier, because you don't know what to expect. And so at DukeNUS, then we started working on pathogenesis in various ways to try and understand why people fall sick and what protects us from the next infection.
So this, from what Ashley and you have said, this is a relatively new medical school.
Yes.
So this building we're in is new. It was built, and there was no medical school before?
There was. So we had one medical school up until 2005. And the medical school was fashioned after the British system. We went to medical school after high school.
Right.
But in the late 90s, Singapore decided that they were going to develop a biomedical industry as one of the pillars of our economy. And so when they looked at the biomedical landscape, it was quite apparent that we didn't have enough clinicians doing research. And so then that was the limitation they felt that would limit industries from coming to Singapore. And so they then wanted to set up a medical school that would train clinicians to be conversant with research, these are various stages of research. So when they looked at various possible partners, they settled on Duke. And so DukeNUS was born in 2005. Our first enrollment, I think, in batch of medical students was in 2008, and so we've graduated several batches since. The PhD program started in 2010, and so now we have a MD, PhD, and a PhD program running in the school.
So Monday night I sat next to your dean at dinner, so I had a nice conversation. One of the nicer deans I've known, actually. Not all deans are nice.
Good to know.
So yeah, he said you wanted to make it different from the British, make it more like the US model. But was it so important to have a partner, you think? Could you have done it on your own?
I guess a partnership with Duke was like a springboard, right? That you quickly can get up to a certain capacities and with a reputation and all that. And we imported the curriculum from Duke, and so we didn't have to reinvent the wheel. And Duke had a 4-year MD program, but with a 3rd year dedicated to research, and so that was attractive to Singapore.
So is this a permanent liaison, or is it reviewable every so many years? And
so I think the partnership is I don't think there's an end date to it. No. But our funding comes in 5-year cycles. So every 5 years we have this review of our performance, what we've achieved and all that, and the budget is then kind of decided based on what we've achieved and what we are planning.
All right. So we have a little audience here. What do we have? PhD students, some faculty, postdocs, yes? Who works on dengue? Raise your hand.
No.
Half of you.
Go.
You all work on viruses, or maybe not, right? Yeah, all virologists. Who else would come to This Week in Virology, I guess? Right, Dahai, where are you from?
I grew up in China. I came to Singapore exactly 18 years and 2 days ago.
You count every day, huh?
Yes. It's 10th of December I arrived Singapore, 2001.
18 years, and you came to do what?
I studied in NTU for my undergraduate and continued with the graduate study. When I graduated in 2010, I went to US for postdoc, about 4 years, and then I came back. For many reasons, basically it's my best choice after my postdoc. I joined NTU. Again, there's an even newer medical school. In case you don't know, it's called Li Guangqian School of Medicine. It was founded in 2011, 2012, and I came back 2013 as a first batch faculty. Yeah.
So, Eng Eong, you said you wanted to be a doctor, and you got dissatisfied and went into science. And what about you, Dahai? How far back can you remember wanting to do science?
Well, I guess since I was young, it's just out of curiosity, I want to understand things. But I guess that's nothing to do with my determination to do science. To be very honest, even after my high school, I don't know what I want to do.
Yeah.
Coming from a small rural area in China, you don't get a lot of exposure, you don't get a lot of hands-on experience of different area of career, etc. Basically, I don't have any idea. And when I came to Singapore to start undergraduate, back then the reason I chose life sciences is just because I was told that life sciences is the most competitive, it's most difficult to enter. So I just try if I can enter it. And unfortunately I entered, and since then I become a life scientist. And in terms of virology, I started a research program at the 3rd year, again with Subhash. And I remember I visited the DukeNUS, Eng Eong, and only 4 or 5 of them are around, and start to know more about virology. And it's just elegant. You can call it species, right? And small, but can do so many things, and just get more and more fascinated. During my postdoc, although it's not directly with virology, but it's the other side of the same story. We look at the host defense mechanism of antiviral defense mechanism. So, and then since I come back to the same community with the same friends and colleagues, very naturally I end up being a structural virologist looking at the positive sense RNA virus in general and of course dengue. Yeah.
Do you often come here to visit, to talk to collaborators?
As much as I can.
Yeah. How far is it?
35 minutes, around 35 minutes. This morning take about longer because running a jam, but yeah.
So tell us a little bit about Nanyang Technological University. Did you say there was a medical school there?
Yes, so Nanyang Technological University itself is young. It's 20 plus years old, and one of the youngest university, top ranking, if I may say that, according to some ranking systems. But and then medical school is even younger, so it started 2012. Again, I think is the government want to train more clinicians, and they see the need of Singapore for clinicians, for doctors. And very similar to DukeNUS, the Lee Kuan Yew School of Medicine is also a partnership between NTU and the Imperial College London Medical School with similar motivation, and probably we see the success of DukeNUS and we want to copy the success and make it similarly good medical school. And so slightly different from Duke, we are UK model, very similar to the National University of Singapore's medical school, 5 years curriculum, undergraduate medical school.
How many medical schools are there in Singapore?
Three.
So
if I may add, so the reason why we have DukeNUS was to supplement the biomedical industry, at least to fill a gap. Then about maybe 10, 15 years ago, I mean, the population demographics became clear that Singapore was going to face an aging society, a problem with increasing proportion of aged. So that the third medical school was started because we felt that we needed more doctors to deal with the problem. So altogether, I think each year we graduate about 500 doctors a year, and so the ratio of the population, the doctor population ratio has changed quite tremendously over the last 20 years or so. I think at one point it was about 1 in 600. Now it's down to about 1 in 400, 1 in 300.
One doctor per 400 people. Yeah. So Lin Fa used to tell me he had to go back to Duke often, like once a year. Is that right? The three of you have to go back now and then?
I think we all have some appointment in some department at Duke and try to go back as frequently as we need to, either to collaborate. We also have conferences that are set up between Duke and DukeNUS. I think it's every 2 years. And I think Lin Fa does have a requirement to go, although I'm sure he enjoys it.
So some of the faculties have a joint appointment. So we have some faculties who are maybe 80% in Duke and 20% here. And then I am 100% here. So I do not have appointment in Duke. And I think she has some sort of appointment.
An adjunct appointment.
Adjunct.
Yeah.
And
so 100%.
You are also 100%.
No, I don't. Yeah.
So you don't have to go to Duke at all?
No. Sometimes I go to North Carolina to visit my collaborator. I don't go to Duke, although it's just opposite.
Got it. Okay. How often do you go?
I go 2 or 3 times a year, but I have family in the area as well, so I always make sure I visit Duke when I'm in town.
This is my first visit to Singapore. It's the longest flight I've ever taken. It was San Francisco to here was 17 hours. And it was also getting to San Francisco. So it was like 24 hours of flying. I don't know how you can do it, but Lin Fa said you get used to it.
There's direct flight from New York to...
There is a direct flight.
No, there's a direct
It's
flight.
more
pleasant,
expensive.
probably. Yeah.
And they wouldn't pay for it. It's okay.
Yeah.
I don't mind. Get more miles. The direct flight is on Singapore Air.
Right.
And I fly United, but it's fine. Going back is a little faster. So let's talk a little bit about science. So I just tell you, I read a couple of each of your papers, which is what I do usually to prepare for podcasts, and I want to talk about some of them. And so let's start with you, Ashley. she had this very nice JCI paper
Yes, thank you.
on, I used to be a JCI editor. Would have been fun to handle that for you.
Oh,
I didn't get much virology when I was on JCI.
Really?
And they brought me on to try and increase the
Increase it.
virology submissions. I'm not sure I did, but I'm happy to see that. So it's dengue virus elicited tryptase induces endothelial permeability and shock. So why don't we first start on a little bit of background on dengue pathogenesis.
okay, sure. So, of course, most people who have a dengue infection actually have a short febrile illness that quickly resolves, and some people are even asymptomatic. But, of course, it's a problem because those rare few people who end up having severe symptoms can have bleeding that can, and vascular leakage leading to things like pleural effusion that can be very dangerous, organ failure, and occasionally it can lead to shock and even death. So we are worried about this smaller percentage of people, which is about 5%, or it varies from location to location, that end up with very severe vascular leakage as a sign of disease.
So when we say dengue hemorrhagic fevers, is that the milder form?
So the dengue hemorrhagic fever is the severe form.
Okay.
It's also called severe dengue. They've tried to reclassify how it's termed
Right.
in terms of clinical diagnosis, but those are basically the same. And then you have mild dengue, so people who
Right.
have infection and recover.
And what's dengue shock syndrome?
Dengue shock syndrome is the very worst case scenario for
Okay.
dengue hemorrhagic fever, and those people actually develop shock. So they will lose their pulse and lose blood pressure and need basically intervention and management.
Okay, so there you have extensive vascular leakage,
Right.
right? Now, can this all happen with the first dengue infection, or do you need the second to get this?
Right, so this is a very interesting aspect of dengue pathogenesis. So yes, it can happen during a primary infection.
So you can go all the way from nothing to DSS.
Yes, you can.
Okay.
Some people can have very severe primary infection, but it is more likely that you will develop a severe infection if it's a secondary infection. And that's because there are 4 different serotypes of dengue. So you have an immune response to one, you'll be protected against that one only. And you won't be protected against the other 3 serotypes of dengue. So when you have a secondary infection with a new serotype of dengue, say you had dengue 1 the first time, the next time you may have dengue 2, you get a situation where you may have more severe disease. And there's a lot of debate why that's the case, but it's thought that the antibodies that your body develops during the primary infection are leading to enhanced infection, enhanced immune activation during the secondary infection that results in more severe disease. And that also kind of points to this aspect that your immune system is probably important for this vascular leakage that occurs during severe dengue.
So just parenthetically, I once had a guest on TWiV who actually, she's in the US, but she got dengue while working somewhere overseas, and she described it which was perfect to have someone on TWiV describe the progression. She had a lot of joint aches,
Right.
and she still has sequelae, even though she didn't have DSS, but she still to this day has, she says she has neurological events
surprising, yeah.
that she never had before. And
No, actually, it's very common in adults who get
Yeah.
dengue. That and the literature is very it hasn't really recorded this very well. So, with very few papers I've published on this topic, somewhere between 5 to 30% of dengue patients actually go on to have this very chronic, long-term sequelae, either in the form of tiredness. So here in Singapore, we had a colleague who was a surgeon who got dengue, and for months after that, he could not operate on the same kind of number of patients he would normally operate
because
on.
of joint issues?
No, just tiredness. He just could not concentrate. Felt it's just not himself. In the early days, Benjamin Rush, when he described the dengue outbreak in North America, he suggested the name break heart fever instead of break bone fever, because besides the joint pain, there's this outcome where a certain proportion of dengue patients go on to have this very long-term chronic psychological sequelae.
And she said it's episodic, interestingly. She's fine for long periods, and then she'll have joint aches, but then she has cognitive issues that seem to be related because she never had before. Anyway, I think she was working, I don't remember where, but she had put mosquito repellent on, and she said during the day I was sweating and it was coming off, and then I felt the mosquitoes start to bite me. So she had a great description of it. Okay, anyway, all right, so that is the pathogenesis, the range.
Right.
And the second, this is called ADE, right? Antibody dependent.
Right.
So that's a theory that's...
It's a theory that
That's
there's
right?
a little bit of support for it. Certainly, you can get it to occur in vitro. So if you have cells in a tissue culture plate and you add antibody to dengue, particularly a weakly neutralizing antibody, then you can add dengue as well, and you can get enhanced infection of those cells in the presence of antibody.
Okay.
So it's been proven in that way. It also works in animal models. Where you can inject an antibody into an animal and then give a secondary infection with a different serotype of dengue. You can also have an increased viral titer in the serum and in organs. Yeah, so that can happen.
But that's not what you work on, right?
I do some, because it's so central to the pathogenesis of dengue and our concept of dengue, we usually try to look at both primary immune responses to dengue. I'm really an immunologist, so I'm always interested in what the immune response is. And we also look at secondary immune responses. So they can be very different, and it's also important sometimes to have this aspect of the heightened infection that you can get in animal models and achieve with the addition of antibody, often in the context of immune compromised models. Mm-hmm. Although because I'm an immunologist, I really like working on immune competent models where you can get also a little bit of an increase if you add antibody to the system.
Okay. So now tell us how you got onto the tryptase, which is a kind of protease, right?
Yes.
Tryptase.
Yes, okay, so it all started when I first came to Singapore, so kind of going back to the beginning of the story. I had studied mast cells, which are these really beautiful cells that are granulated. They make very dense granules that are packed very tightly with mediators that are vasoactive. So some of those mediators are things like heparin, which you may know as an anticoagulant, and then these proteases such as chymase, which is angiotensin converting enzyme, so it affects the coagulation, and tryptase, which is an enzyme that can also affect coagulation, particularly fibrin deposition, and it also can cleave tight junctions between endothelial cells. So these mediators are pre-stored. Often when we're studying cells, we try to do transcriptional responses. We want to know what they make as soon as they have are triggered by a virus or infected by a virus. But in the context of mast cells, they're usually activated by stimuli and then they degranulate. It happens within seconds and they release all of these mediators. And so for people who have studied mast cells, you may know them better associated with allergic diseases. So mast cells are associated with anaphylaxis, with allergy, with asthma. And when I first came to Singapore, I had studied mast cells, and then I noticed that a lot of these signs and symptoms of dengue that people were talking about are all things that seem to me to happen frequently during allergic conditions. So rash is a very characteristic manifestation of dengue in almost all patients, and that's also caused very frequently by mast cell activation. And then things like vascular leakage. We know that mast cells line the blood vessels, and they have a very tight association with them. And we thought maybe some of these factors are involved in potentially increasing the pathogenesis of dengue, and particularly in relation to influencing the vasculature, influencing hemorrhaging.
So the mast cells they must be in larger vessels, not capillaries, I guess.
Oh, they are. They're in capillaries as well.
They don't occlude the blood flow, though.
They what?
They don't occlude the blood.
They don't occlude the blood flow. Okay. There's some suggestion that they might reach some processes into the blood vessels on occasion, but mostly you see them sitting really tight up against the side. They're lined up at kind of regular intervals, and they're very beautiful. They're they have these large granules, and it's very nice to look at them in the microscope.
Okay, so how did you make a connection?
Okay.
You had said that some of the symptoms were the same.
Yeah, so we thought we'll just test it as a hypothesis.
Right.
We have mice that are mast cell deficient. We can also look in cell culture. We can add viruses to the mast cells and see how they respond. So that's the first thing we did. And we saw that they just degranulate very dramatically, within seconds, actually.
Degranulate means they're releasing
these granules, yes, into the cell culture media, so you can see them coming out. You can actually watch it and it's very beautiful. They'll suddenly be pouring into the cell culture media around the mast cells as they're activated. And then we also used our mast cell deficient mice. So we have these mouse models of dengue infection where we inject dengue. There are multiple models. So we like wild type mice. We also use immunocompromised mice. And what we saw is that the mice that lack mast cells, or if we gave them drugs that were designed to stabilize mast cells, and that means to block the release of those granules. In either case, we saw reduced vascular leakage and reduced severity of disease. And I should point out that's without affecting the virus itself. So it wasn't something that was dependent on the changing the replication of the virus, but actually blocking the immune activation by the mast cells.
So you don't need dengue replication to degranulate?
No, we don't. It's really based on the contact. We don't know the receptor. That's something we're working on. But it's based on the contact with the cell surface.
Virus contacts and it degranulates,
Yes.
right? Interesting. And so how do you measure vascular
Leakage?
leakage? Yeah.
That's a great question. So one way we do it is the same way it's done in humans. So in humans, they use a value called hematocrit, which is the packed red blood cell volume. So you can imagine that as you have loss of fluid from the circulation, the percentage of your blood that is red blood cells will increase. And so we see an increase in the hematocrit when we have vascular leakage. We also use a method called Evans blue dye leakage. So we'll inject a dye into the circulation, and then we'll measure it as it enters into tissues.
Okay, so mast cell deficient mice, you infect them with dengue, they don't develop this vascular permeability.
No, not to the same extent. You might see a little bit, but really
Okay.
it's not to the same extent.
Okay, so what did you do next?
Right, so then the next question, which I think leads to this publication that you brought up, was really understanding what product that mast cells make could be doing this. And we thought about it, so as a new investigator, I have to kind of strategize what could possibly be the case rather than guessing and trying all different things. And so we thought, if we are going to spend some money and do some experiments, maybe a good way to start is with these proteases. We know proteases chew up all kinds of things. We know they influence coagulation, and we know that they can have been shown in some cases to break tight junctions, which are the links between endothelial cells. So that was the first hypothesis. And so we did various things to study them. And one thing we did, we used some cell culture methods of using human endothelial cells and then treating them with the products of mast cells and with the proteases themselves. And basically all of these experiments led to the point that while other products can influence the leakage, the one that seemed to affect it the most was tryptase. And tryptase, as I said, is an enzyme that causes tight junction cleavage and also affects clotting of the blood. And so there's some drugs that are available that we could use. And we decided to, first of all, test whether drugs could block the vascular leakage in the same way. So that was very effective. So we have a drug called nifamostat mesylate. It's a very specific tryptase inhibitor. It's an interesting drug because it's used in Japan to prevent intravascular coagulation, but it's not used anywhere else. So that was the first evidence that perhaps tryptase is involved. But we also wanted to test in humans whether we saw any correlation between tryptase levels and the severity of disease. And so this is actually harder than you would think because it's very difficult to get well annotated patient samples where you know how severe the disease was and at the same time have some severe outcomes, because usually if people are in a really good healthcare system, they may have interventions like fluid replacement and things that can change the results. So we ended up with 2 sets of human samples. One of them was from Indonesia. It was very old and it had some very severe patients. And those were collected by our co-author, Dwayne Gubler, when he was traveling around. They're maybe 50 years old. And so we decided to test those samples first. And we saw that indeed, there was a very strong correlation between the levels of tryptase and the severity of the disease.
What are these serum samples?
These are serum samples, yeah. And in fact, when we looked at the grades of DHF, so DHF is graded according to severity as well. So very mild DHF will just have some signs of of hemorrhaging, whereas a grade 4 DHF, you'll have shock and no pulse pressure and things like that. So we had serum samples from patients with different grades of DHF, and then we were able to measure the levels of tryptase, and we saw a really nice correlation between the severity of the disease they had and the levels of tryptase in their serum.
So would you like to get more samples locally that you could do this with?
Right, so what we did then, we actually took some samples from Sri Lanka, from our collaborators there, because they had a very severe outbreak, and so they really had enough samples to prospectively recruit. And we did the same thing, we repeated it, and we saw very consistent results in those samples. Of course, we do have studies here in Singapore as well. But we tend to recruit patients much more slowly for those studies.
How many dengue cases a year are there?
This year, 15,000.
15,000. And they range from mild to severe, I guess. Right?
Mostly mild. If you, by what Ashley has said, we don't see a lot of cases with shock and all that, which is why she has to turn to Indonesia and Sri Lanka and other countries to do her study.
So could you give patients an inhibitor of tryptase?
That's a great question. And in fact, we are doing a clinical trial to see if, so we started with a drug, with a mast cell stabilizing drug. That was the first drug that we showed in an animal model worked. And it's called cromolyn sodium. So you might even see it in the US, it's approved for use. Actually, I should say we changed our drug to a drug called ketotifen that's approved in the British system. It's a little easier here. But in general, they work the same way in terms of mast cell stabilizing. And you can see that it's even approved for use in countries. You can buy it as like eye drops. And occasionally it's used to treat asthma, but I think they're moving away from its use. So it's been around for a long time. We thought it's a safe drug, and we decided to test it in dengue patients. So the study we're doing in collaboration with the National University Hospital, which is on the other side of Singapore. The PI is Paul Tembaya, who's a clinician and recruiting patients with us. And and so we've recruited 109 of 110 patients. So we're hoping that in the next few months we'll have some answers regarding whether that drug works and whether we can block vascular leakage.
So these are patients that don't have dengue yet, but
no, they have dengue.
They have dengue. And you're collecting specimens from them.
Right.
And you can annotate their severity, right? So you'll have that information.
Yes. It's monitored very well what their severity is. And basically we're trying to show that we will see less fluid leakage in them. So we're doing quantitative measures of vascular leakage. So
what do you think? Do you think this tryptase is the main contributor, or do you think there are other things that are...
So that's a good question. I think tryptase is extremely important because it does cleave the tight junctions. And I think in addition to allowing the fluid to leak itself, it probably increases the accessibility of other inflammatory products to the vasculature as it's doing that. Certainly, we think there are other things that are involved. There are leukotrienes and prostaglandins have been shown in some studies to be quite important as well in vascular leakage. And then there's a whole other maybe side of the literature that's not so much what I do, which looks at the potential of viral products to influence the vasculature. And there's indication that possibly some of the dengue proteins itself, such as NS1, might induce vascular leakage. In our system, when we give tryptase inhibitors, we're able to block almost all of the vascular leakage. So we think it's really quite important as a host mediator for allowing this to progress. But certainly there are other things.
Yeah, we've done some papers about NS1 permeabilizing tight junctions. I understand there's a little controversy there.
There's controversy, yes.
So it would be great if it were the main agent, and then you could treat patients, right? Now, do you think it would modify symptoms all the way up to a DSS, or there you have different things playing into it?
So that's a very difficult question. Yeah. So I think that a lot of times it's a cascade. So with DSS, the real issue is that you've lost a lot of fluid volume from the blood, and you're not able to pump blood efficiently. Yeah. So I do think that it's a cascade of events, and probably begins early on, such as these time points that we talk about, where when we have initial immune activation could be quite important. But it's always possible that there are other processes that are end-stage processes that are initiated later that may also be important.
So if you see a good outcome in this trial, what would you do next? In other words, treatment does ameliorate symptoms.
Right. So, on the one hand, we're interested in mast cell stabilizers, which I talked about first. That's what our trial is on, and we think that they might need to be given quite early to prevent the activation of mast cells, because mast cells initially respond to the virus, and that perhaps down the line we should develop some of these other things like the protease inhibitors that might be able to work a little bit later in the disease course. So kind of touching on your previous question, once they've actually been released, can we inhibit them and can we inhibit their function? So I think trying to understand what is the correct time point to administer a drug if it's effective, and when it would work and when it might not work would be some of the next stages for the development.
So the question just left me. What was it? A senior moment here. I forgot. Anyway, so there it is. Came back. Do we have early rapid diagnostics for dengue where you could catch it early? Because you say you need to do that.
So that's another aspect of what we're trying to do. So on the one hand, we do have diagnostics for dengue in the sense that we have NS1 tests, for example. So you can test for NS1, which is a viral product in the serum, and then you can say, okay, yes, this person has dengue. But what we don't have is any test that could tell whether that person is likely to progress to severe disease, if they're going to develop DHF, DSS, severe dengue, and really need intervention. And that's important in the context of an outbreak when there are a lot of patients that have dengue. How do you decide who you keep and who you can send home? And who needs fluid replacement that will need it, that maybe you can't identify yet. And so in that case, we're trying to develop biomarkers of severe disease. And we think some of these mast cell products, as I mentioned, tryptase is correlated with the severity. We have some other products that give us a little bit better test results in the sense that they're easier to measure. And they also are mast cell products that give us a nice correlation with the severity of disease. And so we're trying to develop those further and really prove that perhaps if we test people and try to measure these markers early in the disease course, we can keep those patients that need more monitored treatment.
So what is it that makes patients go to get care when they have dengue? What is the symptom that sends them to the ER or wherever?
Maybe Eng Eong would be better at answering that question, but I think one point is that it varies in different cultures. So like what might send someone here in Singapore is very different from what might send them to the doctor in Sri Lanka or in Indonesia or some of the other places we study, and that factors into how we study disease as well. I don't know if you have something to add.
Yeah, no, usually it's the fever that sends them to
fever.
So high fever, this intense body ache, joint pains, headache. It's a lot more prominent than many other viral illnesses. In Singapore, because most of the cases occur in adults, then they need to get a medical certificate to get off work that day, and so that's how we recruit our patients. And in other countries where dengue occurs mostly in children, then most will try and self-medicate and see what they can do at home first, and then before they seek medical attention.
So fever and aches, do you think that would be too late to give them a tryptase inhibitor?
Probably not. Okay. I think there's a classification scheme now, and like WHO has a protocol for monitoring for clinicians, which I'm not, but I've read it, and it says that basically you monitor them for certain warning signs. And so the problem with that warning signs that they have is they're very good at predicting severe dengue, but they can come up suddenly. And right now it's about 6 hours or so that I think is the window of time when they basically have to intervene between the warning signs and very severe disease. And so the idea is that perhaps we can give drugs earlier, perhaps we can test earlier. And I think right now people don't know that there's something they can take. They know there's not much that can be done, that they'll be sent home and given something like Panadol or Tylenol, which is just to prevent their fever and make them feel a little bit better. But if they knew there was something, we think they might come a little earlier.
I can also add that sometimes the availability of rapid tests is there. You can use it, but the clinicians don't use it because nothing can be done, even if you make the diagnosis, right? The minute you have a drug, then the test actually becomes very useful. It'll be even more widespread in the usage.
For many viruses, you don't even have a test because there are no drugs. And they say, well, why should we develop a test because we can't do anything? So here it's actually good that I'm thinking of rhinoviruses, there's no rapid diagnostic yet. You could make antivirals, but nobody bothers. You can't diagnose it. There, the infection's over in 2 days.
Yeah.
You have to really go quickly. So continuing with dengue, Sheemei, let's talk about your work. So I'm influenced now by Felix Ray, because I just spoke to him recently. So I'm interested in this paper where you're talking about the morphological switch at 2 different temperatures, and I love the bumpy viruses. So tell us about why you're interested in this.
Okay, so dengue virus, it's hard to really make a vaccine.
Yeah.
So everybody knows that in a vaccine, you have to put in 4 dengue serotypes. So there's only 4 serotypes. And the problem, and also these 4 serotypes in the cocktail must elicit equally high immune response. Because if one is better than the other, it actually will cause a more severe disease by antibody-dependent enhancement. So, but even now, if they include all 4, well, I don't think they can always elicit 4 equal. But added to the complexity of 4 serotypes, we see under the electron microscope that the virus can adopt different morphology too. So that completely changes the antigenic structure. So like one antibody that can neutralize the round particle is not good with the bumpy particle, and that could cause partial antibody binding and will lead to ADE2. So it's important to study the different kind of morphology of the virus that exists in nature. So we see all lab-adapted strains. When you grow them in mosquito cell line at 29 degrees Celsius, they look round, and then when you incubate them at 37, they all become bumpy. So, and I mean, does this translate to whatever that is circulating in the natural world? So that's why that leads to that paper.
So let me ask you, if you look at the vaccine preparations, you see a mix of bumpy and smooth particles?
Sanofi didn't ask me to look at their structures, so I wouldn't know.
Okay.
Yeah. So but it is important for them to figure that out. Whether they are putting the virus that is round or bumpy to represent their vaccine, and will it cross-protect against whatever that is circulating in the world. Yeah. So in that paper, we were trying to figure out on the E protein surface which residue is important for causing the virus to be bumpy. So we did individual mutations, and then the final conclusion is it can happen in multiple places, and the substitution can be quite subtle. It's like from a serine to a threonine. It's not that obvious. So just by looking at the primary sequence, you wouldn't be able to predict whether it's a round structure or a bumpy structure, and it can happen anywhere in the E protein. So, and the second conclusion that we make is we look at the clinical isolates. We grow them. So when we get the clinical isolates, we sequence them, and then we passage them in mosquito cell line, and then we sequence them again to make sure there's no mutations. So that we know that we are looking exactly at the same virus that is isolated from the patient. And when we look at it, actually most of them are round particles at 37 degrees Celsius. So that means the lab-adapted strains has gone through, because they have gone through numerous passages. Maybe the bumpy one is, like in the human, when you transmit, they are more likely to select the smooth particles rather than the bumpy one. But in vitro, because there's less selective pressure, you have more of the bumpy ones. They can tolerate a lot more mutations. Yeah. So in nature, most of the virus particle is smooth particles at 37 degrees Celsius, and but there's one strain that is very interesting. So it happens together with another smooth strain, the smooth surface particle. It is within the same outbreak. So what we think could be happening is maybe the smooth particle infect the population first, and then in order to escape from the immune system, then it changes itself to the bumpy surface in order to survive. So although the selection of bumpy surface is not the most optimal in the natural circumstances, but they can do that in order to escape. Yeah, and then we look at 40 degrees Celsius when someone is having a fever, what happens to the virus particle, and they all turn bumpy. So it suggests that for therapeutic, what antibody to use for therapeutics and prophylactic. So in a prophylactic situation where you are predicting that you may get an infection, so you will be injecting yourself with an antibody before infection. So you will likely encounter the virus when it's smooth surface. So it suggests that this group of antibody will be useful for prophylactic. But then when the person comes in with a fever, then for therapeutic antibody, you should use those that are good for the bumpy surface particles.
Mm-hmm. So structurally, what's happening in the bumpy particles? The E tilting up?
So in the compact smooth surface, the E are very tightly packed.
Right.
But then in the bumpy surface, the E to E protein interaction actually loosen. So the E protein actually move outwards, and they are much looser. So the antibodies that are very, very neutralizing to the compact structure, they actually bind across E proteins. So,
Right.
but in the bumpy surface where the E protein has loosened, this antibody, because the distance between the E proteins has increased, they are unable to bind. So those.
I think I learned that from Felix. So the, what is it? The fusion loop epitope, right? Which would be bumpy. It's immunodominant, and those antibodies bind tightly, but they don't neutralize, right?
Yes, in the compact surface, smooth compact. But then in the bumpy one, is a lot more exposed.
Yeah, so that fusion loop is exposed, and that's not good because then the antibodies will bind, but they don't neutralize, right?
No. So the problem with the fusion loop antibody is in the compact surface particle, very few are exposed. So the occupancy that the antibody can achieve is very low, and therefore you get the ADE. But if you have a bumpy surface particle, if the fusion loop is exposed more, then more anti-fusion antibody can bind, and that will neutralize. So I would think the antibody may have a different potency against different flavivirus structure.
Okay. So a single amino acid can cause the transition between the two forms.
Yes.
At 37 degrees.
Yeah.
And at 29,
all are smooth.
They're all smooth, and that's of course the mosquito temperature, probably,
yes.
right?
Yes.
So in mosquito, did I ask you this? I don't remember. Is there a difference in infectivity between the smooth and the bumpy particles?
Not in vitro. So we tested the antibody, the virus, the two morphologies, virus at 37 to viral cells, which is a mammalian cells. It doesn't look like there's much.
Yeah.
Yeah. But the growth rate is about the same, but the thermal stability of the virus is different. So the bumpy surface, if you incubate them longer, like for 37 degrees for like 1 hour, half an hour versus 1 hour, the bumpy ones die quicker than the compact surface. So I maybe it's in the human body, it may be that the dengue virus need to travel a long way to find its host cells to infect. So maybe the thermal stability. Because it stays at higher temperature for longer before it finds a new host cells. So it may die before the smooth particle, and therefore they select more of the smooth particle virus.
Mm-hmm. So this study was done with type 2, right?
Yes.
Does this transition occur with other serotypes?
So now we are looking at the others, and they do have also the bumpy, and then we are looking into it now. The other clinical strains, whether they have a very other structures or just the bumpy and smooth, and different temperatures too. So the thing is, dengue 2 is the widely studied dengue virus for flaviviruses. So, and there's a lot of laboratory strains. But dengue 1, 3, and 4, I don't think there's laboratory strains, are there? Oh, dengue 1 there is Westpac, but 3 and 4 there's none.
They're harder to grow.
Harder to grow.
Yeah.
Okay. Or they don't plaque or something.
So this transition occurs outside of the cell, right? it doesn't happen in the cell. Does this have anything to do with the, so I know during the transit of the particle in the cell as it's maturing, correct me if I'm wrong, it's initially bumpy, or it's
yes, it's another structure. It's not the bumpy structure. It's a trimeric structure.
Oh, it becomes it goes from trimer to dimer, right?
Yeah, yeah.
And then PRM is stuck on until it leaves to keep it from fusing again, right?
Yes.
I got that right?
Yes, you get it correct. Yes.
I'm not used to envelope viruses. The coronaviruses, I purposely chose one without an envelope. It's too complicated.
So, you know, dengue, although it's very simple, there's just PRM and E on the surface.
Yes.
It's very dynamic, the structure. So the bumpy and the smooth, I'm just describing the virus that is extruded out of the cell for the mature virus.
Right.
And because the maturation process is not very efficient, so the virus particle that is extruded out also consists of immature virus, highly immature, and partially immature. Yeah, and then for the mature, you have the smooth and the bumpy. So, and all of them can infect in one way or the other. So that means they are all real pathogens.
Right.
And maybe our vaccine design is a little bit too simple.
So actually you did another study where you looked at immature dengue, right? Which is not infectious. These are particles that are...
the subviral particles.
Yeah, and then there's PRM on it, and you showed that if you put an antibody to PRM, converts it, right?
Yes. So the immature virus, because the maturation process is not very efficient, there are immature viruses that are being released.
That are produced,
Yes.
Okay.
So this immature virus can infect when it is complex with an anti-PRM antibody.
So normally PRM would block it from infection, right?
Yeah, because it caps the fusion loop. So the virus is unable to fuse in the endosome. But this antibody, when it's bound, right, it actually causes some steric hindrance. So the immature virus, when it goes to the endosome, it needs to rearrange from the trimeric structure to the dimeric. So there's a lot of structural change. And because the PRM is decorated with the antibody, so when the E proteins try to change, the PRM, because the antibody give it a lot of steric hindrance, and it's attached very strongly to the PRM. So the PRM doesn't move along with the E protein, and it got knocked off. And so the fusion loop of the E protein is then exposed, and then it helps the virus to fuse.
Got it. And you did structures of these to understand how the antibody is pushing the PRM off, right?
Yes. So we combine cryo-electron microscopy with because it gives us a really low resolution structure, 20-something angstrom. So nowadays, no one believes the 20-something angstrom structure. So we have to supplement it with a deuterium hydrogen exchange method, and also molecular dynamics to simulate how it moves from one stage to the other. Yeah, but together, the structure is so much stronger, and it shows the whole picture of how the E protein can move.
Okay. All right, great. So speaking of vaccination, do you vaccinate here against dengue? No? You don't use Dengvaxia, right?
So Dengvaxia is licensed in Singapore, but it's not widely used because it can only be used in those who have had a prior episode of dengue. In Singapore, the population that starts to have a prior episode, it's around 30, 40 years old. Yeah. Right now, the safety data, it reaches 45 and not above. And so the population younger than that are mostly not eligible for vaccination. So that's why it's not very widely used.
So there are other dengue vaccines in the pipeline.
Yes.
Right. So maybe those would be, because maybe you would need to know the serostatus of the recipient, right?
At least for the phase 3 trial data from Takeda's vaccine, it looks like it works in those who are seronegative as well at baseline. So if that holds true, then at least for Takeda's vaccine, you don't need to know whether they've had a prior episode of dengue. But for Sanofi, apparently you do.
Okay, Eng, let's talk about yellow fever. It's an interesting paper. Metabolic perturbations and cellular stress underpin susceptibility to symptomatic live attenuated yellow fever infection. I think it's cool you use a vaccine strain to study pathogenesis, right? Because you can't use the wild type strain.
That's right.
So tell us why you did this.
Well, I mean, as Ashley mentioned earlier, not everyone who who get infected with dengue virus will get disease.
Right.
And that's quite universal for all flaviviruses, that actually only the tip of the iceberg develop disease. Majority won't develop anything. They don't even know that they're infected. So then a big question in the field has always been, why do people fall sick with the same infection? And so that's something that we've been really keen to understand. And we kind of stumbled on this yellow fever vaccine approach to studying infection rather accidentally. Because I was interested in antibody-dependent enhancement, ADE, and the mechanisms that go along with it. And every time you write a paper, one reviewer will always ask us, is this clinically real? Is this clinically relevant? And so I thought, I'm a clinician, I should solve this, right? So then some years ago, we decided that we have 2 vaccines, both flaviviruses, and both are licensed for use in Singapore. And there's no contraindication from using 2 vaccines at the same time. So we then recruited 84 healthy volunteers who have never had prior vaccination or infection with dengue. We vaccinated 3/4 of them with JE, Japanese encephalitis vaccine, which is related to yellow fever. And then after that, give them the yellow fever vaccine, which is a live vaccine. So if antibody-dependent enhancement would occur, then the antibodies to Japanese encephalitis should enhance the yellow fever infection, right? And as a control, the remaining quarter got just yellow fever vaccine.
Okay.
So we then tracked what happened to these subjects. And true enough, there was ADE, but the ADE was only limited to those with a very limited range of titers. So in other words, suggesting that you need the right ratio of antibody to virus to form an immune complex that then optimally ligates the right Fc receptors. Since that publication, 2 other papers came out for dengue, and these were cohort studies that came to the same conclusion that not everyone with secondary infection are at risk of severe dengue, only those that had a narrow range of cross-reactive antibody titers before infection. But as a result of that trial, we then realized what we could do with yellow fever vaccine, right? About half of them developed symptoms, very mild, flu-like illness, about 6 days after they are vaccinated. Most complain of headache, a bit of body ache that lasts for about a day, and a handful will have fever, right? And so that then gave us the opportunity to ask, why do they why do they develop this flu-like illness when the other half are completely well, right? And so that was a story in how we then, because we took blood samples before we vaccinated them at various time points after we vaccinated them, then we could really dissect the molecular processes that eventually led to a febrile illness.
So then you decided to do transcriptomics on these, right? To see if you could associate something with symptomatic vaccine.
Yes.
Right. So what's the outcome of that? What did you find?
So what was surprising to us was the molecular processes preceded the symptoms by about 5 days. Right. So immediately after vaccination, you see these innate immune responses that correlated with symptoms that occurred 5 days later. So, but again, only half of the subjects responded with this kind of signatures. The other half didn't do anything with the same infection. And all of them, you could detect virus in them. Right? So obviously they were infected, they developed viremia, they developed antibodies, but only half had this unique signature. So because it was so early, we then said there must be something that occurred before we even vaccinated them. And so then we looked at the pre-vaccination samples. And there the most striking thing was that in those that had that went on to develop symptoms, the cells were under some level of stress, or called endoplasmic reticulum stress. So ER stress, very I guess in layman's term, is a factory's effort to exert some quality control on their products, right? So it looked like in these subjects, at least in terms of the bioinformatics, it pointed towards lymphocytes being under some level of ER stress. So what potentially we think could be happening is that they either had a recent infection, or they're responding to some microbiome in their gut, that they were actually secreting antibodies, and that's why the lymphocytes are under ER stress.
Some infectious process, you think?
Yes.
Not just general stress like having to
It could be as well. We
pay a
never
bill or
control
something.
them. Could be lack of sleep, like most of us. So that's so about half of them are undergoing ER stress. And as a compensation, they then alter the metabolism, and that's well known that the metabolism gets downregulated to compensate for the ER stress. So then when they're vaccinated or they're infected with a live virus, then the 24 hours later, the top transcriptional pathway that comes out, the top hit is translation. The second top hit is Krebs cycle activity. So it looks like, and that makes sense because the virus needs energy to grow, it needs to make new proteins, needs to assemble new virions. And so in those subjects that are already undergoing ER stress and compensating by reducing metabolism, this increase in translation and increase in energy demand then tips the whole thing over into a maladaptive ER stress that triggers a reactive oxygen species response, triggers a innate immune and pro-inflammatory cascade that eventually leads to symptoms.
And you think this may also happen in wild-type virus infections, right?
That's our guess. Yeah.
So it's an indicator. So the idea is that, and maybe this is general, that if you have a pre-existing infection or some sort that would cause stress, then the outcome of a different virus infection can be more severe.
Right. Because
it could be any virus. It doesn't have to be dengue or yellow fever, right?
Yeah. As long as I think the infection process is efficient, then likelihood of these events happening increases.
Yeah, this is very interesting because we always wonder what makes the range of responses to antivirus, and the idea that there's some other infection going on or some other stress is really
right.
I don't know how you would avoid that though. It's life, right? You're continually infected.
Yeah. So there may be some truth in this traditional saying, right? And get enough sleep, make sure you're well fed, or look after yourself well, and that's the way to prevent illness.
But as you say, for vaccines in general, the side effects are typically not severe enough to warrant any precaution. But yeah, maybe in general you should stay healthy to avoid really severe symptoms of any virus infection.
Right.
Because you're not going to be able to control it. So that's really interesting. I just wanted to ask you briefly about another study you were involved in. You were second to last author, where you're looking at microneedle delivery of, this was a dengue vaccine. And I always tell my students that this is going to be the delivery method of the future. And in fact, we were talking about anti-vaxxers. I bet if you took the needle away, it would make a lot of anti-vaxxers change their mind. I think that has a lot to do with it. They don't like the kids getting 10 jabs over and over. My kids, I'm sadistic, I guess. I let them get it. But if you switch to a microneedle, maybe we could get half of the anti-vaxxers.
Yeah.
So you showed it was very effective for dengue. So do you think this could be used for almost all vaccines?
Well, the thing that attracted me to this technology, which was really the work of Paula Hammond's lab, was that you could release the vaccines in sequence. As Sheemei pointed out, you need for dengue vaccines now you need to give 4 different viruses at the same time, and one of the challenges with that is that these viruses compete.
Sure.
Right? And so then you get a very imbalanced immune response. Now, if we could give these vaccines one at a time, given what we know about the competition and how we can even perhaps take advantage of sequential infection, where the immune response to one may actually benefit the infection with a subsequent vaccine strain, then you're giving the immune system a chance to see one virus at a time rather than pour in antigens. And some years ago, there was this work from, I think it's the UMass group, that computationally looked at the germinal center where antibodies are made and asked the question, what happens when you give a cocktail of vaccines versus one vaccine at a time? And computationally, the answer is when the maturation process is better when they see one antigen at a time, rather than giving them pouring in a cocktail. And so that was what led me to collaborate with Paula Hammond and ask the question: if we do have a vaccine like Sanofi's or Takeda's, where right now we're pouring in all 4 at the same time, but if we could give them, one at a time, then what's the outcome? But we don't have the technology to give vaccines one at a time, and giving people 4 shots over, 4 times over the course of a month, the compliance rate is going to be very low. Yeah. But if we could engineer everything into a patch, and all you do is give a patch, and the patch then takes care of this sequential delivery. How
many vaccines could you put in a single patch?
We only tested 1, right, because the idea that what we wanted to show as a proof of concept that live vaccines could be delivered this way as well. Because up until now, everything has been on proteins, on subunit vaccines, or even killed viruses. But no one has been able to show that you can preserve a live virus in this method and deliver it at the same time.
Did you say that you'll be able to have sequential release from the patch? So what's the number of sequential releases that are possible, do you know?
Well, according to Paula, our imagination is the limit, because it's all chemistry. And you just have to layer these chemistry one at a time, and it's what's practical.
So you could put 20 different, so some of these bacterial vaccines have 20 different components in them. Right. And so you could do that in theory. Right. So that would be fine for vaccines that work through injection, but like polio vaccines that you take for the gut, you can't do that because it has to be delivered. But we're getting away from those anyway. And by the way, that's why you have 3 doses of OPV, because they compete with each other, and you have to do it 3 times to make sure you get all 3 immune responses against. But I think this is a great technology, and it will, I think it will really encourage vaccination if we can get that. So what's the timeline, do you think, for this coming on board?
Yeah, I guess, like what you said earlier with Lin Fa's answer, is funding that's limiting how fast we can develop this. I think, from what I've learned from Paula Hammond, a lot of work is going on with HIV vaccine and exploring the system as to deliver these vaccines. And because of the thickness of the needle, you can also engineer it to deliver vaccines into where the antigen-presenting cells will be, rather than rely on clinicians to shoot this into the subcutaneous space or intramuscular space. So you get a lot more better precision on where the vaccines need to be.
I can imagine one patch that will do them all, right?
Yeah, that's the hope. That's the
one
hope.
vaccine patch to vaccinate against them all. I'm trying to make a ring analogy, one ring to rule them all. I'm not doing a good job. All right, we came back just in time.
I did.
I was in Texas doing a podcast, and someone left, like you did.
I need to do this.
And when he came back, I said, what happened? He said, I'm an old man. So you're not old.
Yeah.
All right, you came back just in time. We're going to talk about your work now. I know we should be finishing soon, but we'll go over a little. You published this paper, High Resolution Structure, reveals the mechanisms of RNA recognition and activation by RIG-I and MDA5.
Yep.
So tell us about that.
So we were interested in how the host defense mechanism sense the RNA viruses in general. And then RIG-I MD5 was discovered to be the major intracellular sensor for RNA virus infections. As a molecular biologist, a structural biologist, we want to understand how. Basically understand what is the chemistry, what is the structure, explanation of this specificity. We know within the cell there are thousands of copies of different sorts of RNA. Why is the viral RNA? What is the particular signature, either is chemical or structural or temporal spatial features of this RNA that get specifically picked up by RIG-I-like receptors. Obviously, this has to be very specific. Otherwise, you have huge trouble, right? You have autoimmune conditions. So the story was, as we know, is a very hot topic. People like it, and many labs are trying to understand it the same as we do. I remember what I did was to take a shortcut, just try to generalize what are the most common feature that's generated for the viral RNA species. It has to be common across virus, but not from any host mRNA or any host RNA. Triphosphate, as at the 5' end of the genomic or subgenomic RNA that were generated during the replication, is obviously a key feature. And another one is within our cells, we don't have a lot of double-stranded RNAs. And that's the species that was generated also during the replication. Very similar idea of how the specific antiviral works. You really have to target those area or molecular signatures that only virus process, but not the host. So these are the very tiny bit of features that are essential for the receptors to recognize. And we use this kind of RNA species to do our experiments, and it turned out to be exactly the same. And in our study, the key findings are basically proof or explains the basis of how these RNA features, whether it's just a triphosphates at the end, or it's the double-stranded RNA nature that are specifically recognized by Regi. And this specificity are just so tiny, but still essential to differentiate the viral RNA against the pool of host RNA species.
So the 5' triphosphate and double-stranded.
Right, that's the major essential feature that Regi-like receptor recognize. For MD5, there's a slightly different story. It works very similarly to Regi, but complementary. Instead of the extremely 5' end during the replication, it focuses on the whole replication intermediates. The longer duplex RNA, the more significantly it can activate MD5. And that's why these two receptors sort of complement each other without too much overlap, but covers majority of probably every single RNA species that they can recognize.
So for Regi, the double-stranded character is shorter?
Yeah.
But you have to have a triphosphate, right?
Yes. So for Regi, effective activation of Regi, you need both triphosphate at the end, the double-strand nature right next to the triphosphate.
And for MD8, it's longer.
RNA species.
MD5.
For MD5, then a 5' end is not essential because, as we know, many viruses like polio modifies 5' ends.
That's right.
It doesn't really give the chance for this receptor to recognize this triphosphate. Instead, MD5 focus on the whole replication intermediates. The longer duplex RNA, the better. Yeah.
So this paper also provides insight on what happens before and after the RNA binds, right?
Yeah.
So tell us a little bit about that.
Yeah, so in general, my research, we are interested in molecular machines, and those machines are change conformation upon binding to their ligands. And Regi or MD5, the common feature they have is that they have this so-called RNA helicase. RNA helicase domain. It has all these conserved motifs. By many means, it looks like RNA helicase, but they just don't do a helicase job. They don't go and unwind or resolve RNA structures. Instead, they are evolved specifically to recognize this double-stranded nature. And together with accessory domains, which usually happens at the ends of the helicase conserved, this core region. For Regi, they have this C-terminal domain, which evolved very specifically to recognize triphosphate moieties. And this helicase plus this CTD domain, they work together very nicely to recognize the two features at the same time. That generate the high specificity that they have to find this species before they get activation. And then, as we mentioned, during the resting stage, you will never want to activate these innate immune receptors. And the mechanism for Regi to have this resting state is that it's signaling domain when there's no viral infection, when there's no right viral RNA species around, they are interacting with itself. So it has this auto-inhibitory state for the receptor. So that keeps it rest during the, say, peaceful, healthy condition.
So Yeah, because these proteins are everywhere, Rna in all cells.
It's ubiquitous. Yes.
You don't want them to be inducing interferon unless there's foreign RNA around, right?
Yeah, indeed.
So you say the helicase and the CAR domains are interacting and it keeps them repressed.
That's right, yeah.
And then when the RNA binds in the way you mentioned, it opens up. And then the CAR domain can interact with MAVs, right?
The downstream adapter protein
Right.
MAVs is on the mitochondrial surface. And what it does is, Rna is like starting a fire.
Right.
And the MAVs domain is like the woods. It's like the woods on the mitochondrial surface. Once you start this fire, and these MAVs domain will aggregate in a way, form a cluster of proteins, and that gives the opportunity for the additional signaling proteins to amplify the signal and eventually lead to the transcription activation of interferon and the other antiviral factors.
So you also mentioned that MDA doesn't have these repressive interactions, right?
Yeah.
So how does it maintain the off state?
So for MDA5, instead of have tight control of its own conformation, it turned out to be more floppy, right? The domains don't really connect. They are only connected with the floppy linkers, and they don't really interact with each other. And that creates a state that they have a very high threshold to get activated. Only when they are clustered together by the means of long duplex RNA, they have enough number of MDA5, and their adapter, their signaling domain, the CARS domain, has the chance to come close together and get the chance to activate the downstream. So Rna, it needs its self-discipline, self-control. MD5, you need a stronger activation in terms of a long RNA duplexes.
Okay.
Yeah.
Yeah, I've been following this field for a while. It's wonderful how the structure is informing our understanding of how it works. As you would predict, structures, I don't have to convince you, structures tell you an awful lot, right? I want to ask you one more thing. You're also interested in designing RNAs that will stimulate these proteins, I guess as therapeutics, right?
So the idea of designing a RNA species that have the ability to modulate Rna and or MDA5 comes from basically the basic study of what they do, how they function. We know that many times when people develop vaccine, they need the adjuvant, and adjuvants, they are the existing ones. They are very prone to certain types of immune response, but not broadly or not specifically to viruses. We were just thinking, since these are the by default major receptor for antiviral immune response. If we find good ways to manipulate these receptors, we get good adjuvant, or at least potentially a good adjuvant.
I see.
Yeah,
Okay.
so that's the motivation. In terms of antiviral directly as therapeutical antiviral options, I think is very dangerous. Probably we are not smart enough yet to be able to precisely turn or modulate the activity of this receptor so that it has serve the purpose to eliminate the virus without causing too much danger.
Right.
Yeah.
So you're thinking of delivering it with a vaccine and a way to be an adjuvant, as you say.
I think to develop an adjuvant is more reachable than directly using as antiviral.
In fact, some adjuvants are TLR ligands.
It is, yeah.
Right?
Yeah.
I'm thinking of
And then
one that's a
the-
TLR7 ligand or something like that.
Yeah, and then further on this point, the current trend is actually making use of the innate immunity for anti-tumor or cancer therapy.
Right.
And there are more and more studies coming out by activating either Rna or STING, these innate immune receptors, to help fighting the cancer, either by itself as therapeutic options or together with these therapeutic antibodies. So there are a lot of papers coming very recently showing very good and promising results.
And you were able to make a small RNA that efficiently activates Rna, right?
Yeah, so again, this is the benefit of doing structural biology, and when you are able to see it atom by atom, you know what kind of RNA you design.
Right.
You only activate Rna without touching anything else, and there's no side effects or non-specific target within the cell. So that's what we did.
Okay, I want to ask each of you two more questions. Okay, short Ashley, if you had not been a scientist, what would you have done?
Wow, that's a tough one.
Only you can answer.
Yeah, I mean, I don't know. I like to paint. I might have tried to be an artist or something.
What's your medium?
Oil paint. I don't do it anymore. I don't have time. I only do science now these days. But yeah, it's a good reminder to remember the things that are central to your personality and try to keep up with them even though you're busy in science.
I know one of my book co-authors, Ann Skalka, is retired, and she said, now it will give me time to oil paint again.
Yeah.
Do that. Sheemei, if you had not been a scientist, what would you have done?
I will be a chef.
A chef?
I love to cook and I love to eat.
Wow, that's great.
I know I love to watch all the Master Chef and whatever chef program, and very intrigued in how they can make like a food into an art piece. Well, I'm not so sure whether I'm that artistic, but the food that I make is delicious. According to my 7-year-old son, it beats some French restaurant with 1 Michelin star in Orchard Road.
She makes very good chili crab.
Yeah?
Yeah.
Yes.
Eng Eong, what would you have done?
I dreamed of becoming a pro tennis player, but I was never good enough. So I guess I had to do this.
Yeah, okay, you could always try. Dahai, what would you do?
Well, my dream was to be a pilot.
Pilot.
Obviously, it won't work. In China, you have how many, young gentlemen who has perfect body and, to be a pilot. Maybe in Singapore there's a good chance, but I'm already too old. But in reality, I think if I failed, for example, during my study and never get a chance to do a PhD, etc., most likely I will end up as a farmer going back to hometown.
Okay, those are good answers.
So in my hometown, we grow peanuts.
Nice. All right, one more. Ashley, RNA or DNA?
RNA.
Sheemei, RNA or DNA?
RNA.
RNA.
Yeah, certainly, same answer.
RNA all around. Wow, that's good. I always love the different...
I have created a new course based on RNA for undergraduates. Yeah, certainly.
Okay, that's our special TWiV here at DukeNUS. You can find all these episodes at microbe.tv/TWiV. And if you have questions or comments, please send them to twiv@microbe.tv. If you like what we do, consider supporting us financially. You can go to microbe.tv/contribute. You can use PayPal or Patreon to make a donation. My guests today, 3 of them from DukeNUS, Ashley St. John, thank you so much.
Thank you.
For joining us. Sheemei Lok, thank you very much.
Thank you.
And Eng Eong Wee, thank you.
Thank you.
From Nanyang Technological University, Dahai Luo, thank you very much. I'm Vincent Dracaniello. You can find me at virology.ws. I want to thank ASV and ASM for their support of TWiV and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral. Thank you.